Photosynthesis in dry climates. In warm, dry conditions, many of the adaptations that plants make to conserve water reduce the availability of carbon dioxide to the chloroplasts. In C3 plants, adaptation increases photorespiration and greatly lowers the efficiency of photosynthesis. C4 photosynthesis and CAM photosynthesis are adaptations that capture carbon dioxide and then transport it to the chloroplasts or store it for later use, eliminating photorespiration. Now, let's look more closely at each type of plant. C3 plants are found throughout the world and are very well adapted to temperate conditions. Most of the trees and flowers that are familiar to you are C3 plants. The C4 photosynthesis is primarily found in the tropical and subtropical grasses, but is also found in some shrubs and eudicote herbaceous plants. There also are a few important C4 agricultural plants such as corn, sugarcane, sorghum, and millet. Crassulation acid metabolism, CAM photosynthesis, is found primarily in succulents, but not all succulents use CAM. It is found in such diverse plant groups as cacti, orchids, and ferns. The important agricultural CAM plants are pineapple and agave. The primary difference between C3 photosynthesis and C4 and CAM photosynthetic pathways is the process of carbon dioxide fixation. As you recall, in C3 photosynthesis, carbon dioxide is captured by the Rubisco enzyme and added to RUBP inside the chloroplast. This produces 3-PGA, a 3-carbon compound that gives the C3 process its name. In C4 and CAM photosynthesis, carbon dioxide is fixed in the cytoplasm of the mesophyll cells. Carbon dioxide enters these cells by diffusion from the stoma. C4 plants have a special carbon acceptor molecule known as phosphoenol pyruvate, PEP. PEP carboxylase, a highly efficient enzyme, reacts with carbon dioxide and PEP to produce oxaloacetate, a 4-carbon compound that gives C4 photosynthesis its name. The oxaloacetate quickly is converted into aspartic or malic acid, both of which are also 4-carbon molecules. C4 photosynthesis uses an ATP molecule to fix the carbon dioxide. This energy expense is compensated by the reduction of photorespiration in conditions limiting the availability of carbon dioxide. This use of ATP in the C4 process makes the C4 pathway more efficient in conditions with little photorespiration. The 4-carbon molecules enter chloroplasts, where the carbon dioxide will be released to be used in the Calvin cycle. Both the light reactions and the Calvin cycle are the same in all types of photosynthesis. The enzyme Rubisco fixes carbon dioxide within the chloroplast. As opposed to C3 photosynthesis, C4 photosynthesis and CAM photosynthesis first fix carbon dioxide into 4-carbon compounds in the cytoplasm of the mesophyll cells. Lowering the concentration of carbon dioxide inside the leaf by fixing it into 4-carbon molecules creates a steeper concentration gradient, which increases the diffusion of carbon dioxide into the plant from the atmosphere. This permits C4 plants to have smaller openings at their guard cells, thus reducing water loss. While both C4 and CAM plants fix carbon dioxide into 4-carbon compounds, C4 plants have important anatomical adaptations not found in the CAM plants. In C3 plants and CAM plants, chloroplasts are scattered throughout the mesophyll, with large numbers found in the long uppermost cells called palisade cells. In C4 plants, the chloroplasts are concentrated in the vascular bundle sheath cells. The 4-carbon molecules produced in the mesophyll cells are transported to the chloroplasts in these cells, where they are broken down to release carbon dioxide. Actively transporting carbon dioxide into these chloroplasts raises the concentration of carbon dioxide inside the chloroplasts and eliminates losses to photorespiration. This process also speeds the entrance of carbon dioxide into the leaf, allowing the stomata to open less, resulting in less water loss. Cam plants do not have the anatomical adaptations of C4 plants. In fact, their anatomy is comparable to C3 plants. The cam adaptation fixes carbon dioxide at different times rather than in different locations. In cam plants, the stomata are closed during the day. How do cam plants obtain carbon dioxide if they keep their stomata closed? They open their stomata at night to allow carbon dioxide to enter the plant. By only opening the stomata at night, when evaporation is the lowest and relative humidity is the highest, cam metabolism is the most water-efficient of the types of photosynthesis. Once inside the plant, the carbon dioxide must be stored for later use. Cam plants fix carbon dioxide into 4 carbon molecules, just like C4 plants. These 4 carbon molecules, such as malic acid, are stored in the vacuoles of the cell, while the guard cells are open at night. Once the sun rises, the guard cells close the stomata, and no more carbon dioxide will enter or leave the plant. During the day, the stored 4 carbon molecules will be broken down to release carbon dioxide to be used in photosynthesis. To summarize, the primary difference between C3, C4, and CAM photosynthesis is the initial fixation of carbon dioxide. The C3 pathway is the most energy-efficient form of photosynthesis, but losses to photorespiration in warm, dry conditions reduce its efficiency. C4 plants have the advantage in these conditions even though they spend more ATP per molecule of carbon dioxide. C4 plants have anatomical adaptations that concentrate chloroplasts in cells into which 4 carbon dioxide carriers are actively transported. This increases the flow of carbon dioxide into the leaf, allowing C4 plants to have smaller stomata openings. This in turn decreases water loss. CAM plants adapt to harsh conditions by storing carbon dioxide in organic acids during the night and releasing it during the day. The CAM pathway is the slowest and most inefficient in converting the sun's energy into glucose. However, it is the most water-efficient of the 3 types of photosynthesis. Thus, it is most often found in desert plants.